Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show. Today, bringing you another really awesome guest involved in science and technology that's creating a better tomorrow for many people out there. Today, we are joined on the episode by Dr. Bill Liu, who is a professor of radiation oncology, a member of the Stanford Cancer Institute, a member of Stanford's molecular imaging program, as well as of the Stanford BioX Interdisciplinary Biosciences Institute. Dr. Liu is a physician scientist, radiation oncologist, and bioengineer who directs both the thoracic radiation oncology program, and he's also a principal investigator of what's known as the Flash Sciences Lab at Stanford, which is this program of, it's focused on the development and study of this really interesting, extremely rapid form of radiation therapy. Ultimately, it's going to be focusing on optimizing the biologic therapeutic index of cancer radiotherapy as it's currently performed and as the whole technological infrastructure behind it. Dr. Liu's clinical research is in the area of clinical trials and implementation of new treatment techniques for lung cancer, as well as the development of novel imaging methods for measuring organ function, as well as predicting a patient's response to cancer treatment. He's also the developer, and he has a startup company that's focused on the technology behind Flash, known as Phaser, which we'll be getting into. Dr. Liu received his medical degree from University of California, Davis, did a PhD in bioengineering at UC San Francisco and Berkeley, and then he completed his radiation oncology residency training at Stanford, certified by the American Board of Radiology in Radiation Oncology and fellow of the American Society of Radiation Oncology and American College of Radiology. A lot to get into today, a lot of really cool topics. Honored to have him. Dr. Bill Liu, thank you so much for taking time to come on the show.
Yeah, thank you so much. I really appreciate the invitation, and thank you for that very kind introduction.
Yeah, no, I'm really honored to have you. I'm looking forward to a lot of these themes that we don't normally touch on in this way. But I would love to start off sort of earlier on in your career. I mentioned you did your medical degree, but you also did your PhD in bioengineering, which is sort of the first place sort of radiation shows up, but not in cancer. But you were in your PhD, you were studying X-rays, actually looking at exocrine pancreatic cells and quantifying the protein content. Really hot topic today in terms of all the single cell analysis stuff that's going on. Talk a little bit about yourself, if you would, and a little bit about putting these two disciplines together at the beginning of things.
Yeah, thank you. So I think that is a theme in terms of putting disciplines together. That's something that is an approach that has kind of fit my interests and skill sets. I enjoy, taking an approach from a physical sciences type of standpoint, but applied to medicine and biology. And so that kind of interdisciplinary thinking and training kind of weaves its way through my kind of personal history. So as you said, I studied biophysics as an undergrad. Then did a year of research at Lawrence Berkeley National Lab, that's where I started doing this research in X-ray microscopy, which at the time was very new. It was using the synchrotron X-ray light source at Lawrence Berkeley National Lab. So that was still is very advanced tool. that was, just starting to be brought to bear on biological problems. And so that I think really fit my interests and got me kind of going on this path of multidisciplinary science, understanding the physics and optics of that, and then applying it to biological research problems. And I'd also always had an interest in medicine, and so pursued my medical studies kind of in parallel to doing a PhD program in bioengineering. And as you said, at that time, my research really was not, related to cancer. It was related to applying this technique of X-ray physics, X-ray microscopy to studying physiology in cells at a single
Hmm.
cellular level. And at that time, I thought, this is going to be a tool that revolutionizes science, and was very interesting to me. As it turned out, I think because of a number of logistical issues of requiring these large facilities and so on, that field I think remains active, but never took off in the way that I envisioned back in those days. But I learned some very important lessons and skill sets about how to do research and about how to kind of take an interdisciplinary mindset to problems. And then developed this, interest in cancer as part of my medical studies. And there's a story behind that. I kind of discovered radiation oncology accidentally, but kind of it was like the perfect fit of technology, science, physics applied to medicine, and having a big impact on a problem that is, really a huge challenge in our world today. Cancer has become, the one, number one single cause of death worldwide. Between number one and number two, kind of depending on how, diseases are categorized and grouped, but clearly, a huge epidemic problem and growing because of the aging of the worldwide population. Um, if you look at the impact, for example, that obviously huge impact of COVID-19 when the pandemic first came on, and in the first 2 years of the pandemic, deaths shot up dramatically. It became very rapidly became one of the leading causes of death worldwide. And in the first 2 years, there were about 6 million people dying of COVID-19. But if you look by comparison, not to diminish that at all, but every single year, 10 million people are dying of cancer around the world. So it really, cancer dwarfs actually all of the communicable diseases. You can add them all up. Again, not to, diminish any of them, but, it's just a matter of scale, right? TB, AIDS, Ebola, COVID, all of those added together, cancer is a far larger source of mortality. And within the next couple decades, the number of people, you know, the incidence of cancer and the deaths from cancer will approximately double over the next, 2 to 3 decades.
Mhm.
So it's a huge problem. It's growing, and so it's a worldwide health crisis, really.
It's yeah, I mean it definitely, as they say, continually, it should be categorized as a significant unmet medical need. And as you're
Yeah.
just pointing out, although we've come a long way, still a long way to go, especially with a lot of these solid tumors and the progression-free survival numbers that we currently see as we sit here in 2025. um, I'd like to maybe go back and start out in sort of the turn of the millennia. So, you do your PhD in 2000. I believe it's a couple of years later, around 2002, I saw your first paper paper where cancer and radiation comes up, and it was in a paper that you published on Hodgkin's disease at the time, and something known as intensity modulated radiation therapies. And in this paper, you're talking about how, in 2002, okay, we're looking at these trends. We want smaller treatment volumes, lower doses per treatment, and we'll get to phaser in a little bit because there's some parallel there. I was wondering if you could sort of take us through, in the 20 some odd years that has happened since then that you've been immersed in radiation oncology. What some of the big trends have been? Obviously, we haven't done much in terms of the technologies on the show. We've done a little bit in terms of the radioactive drugs, the radionuclides, things like that, which is its own little area of radiation oncology. But talk a little bit about sort of some of the trends that you've seen as you've been practicing and these technologies have been evolving alongside. And we'll talk about phaser and flash in a little bit.
Yeah, thanks. I'm impressed by the depth of homework you've done. That's really fantastic. And it is something I think is really exciting because, over the last 20 years, cancer therapy overall and radiation therapy have, evolved dramatically. One of the things that I like to tell the residents that I train is that since the time that I did my residency training back in the early 2000s in radiation oncology to today, there's actually not a single technique that I use in the clinic today, whether it's simple or complex, that's done the same way as when I was in training.
Mm.
And so all of that has evolved dramatically. So I tell them, that's why I think when we're kind of learning radiation oncology today, it's important to learn the specific techniques and be expert at them. But it's more important to learn the general principles because those techniques are going to change. If we're doing anything the same in 15 years as we're doing today, I would actually see that as kind of a failure of our field because it means we're not progressing, right? So to your point, so what's changed? A lot has changed in the last couple decades. You mentioned intensity modulated radiotherapy. That is a technique for really sculpting radiation doses. And so, the ideal that we would like to achieve when we're treating cancer with radiotherapy, radiotherapy is what we refer to as a local therapy. In other words, it's directed spatially at specific targets. And so in that sense, it's very anatomically based. It's kind of parallel in many ways to surgery.
Sure.
And so the ideal, radiotherapy is one where we give a high dose of radiation to the tumor. I mean, that's the goal, right? Is to eliminate the
Sure.
cancer. But all of the organs, the normal tissues surrounding the tumors, we would like to spare from radiation. So what we want to do is create these sculpted radiation doses, so it's like three-dimensional sculpting, and focusing the radiation dose where we want it, minimizing the, spill of radiation, if you will, from reaching other nearby organs.
Mm-hmm.
And that's kind of where a big part of the focus of radiotherapy has been in the last 2 to 3 decades is constantly improving that spatial precision and accuracy. And there's a few things that have contributed to it. One is the what we might refer to as the targeting or target delineation aspect, being able to see what is it that we really want to target. And that imaging has played a huge role in that. And so imaging has dramatically improved or advanced in the last couple decades. CT imaging, MRI, positron emission tomography or PET imaging. All of those have really contributed to our ability to kind of hone down and focus on where exactly is the cancer that we want to treat in the body. And so that helps us kind of define our targets. And then being able to focus the radiation on those targets so that dose shaping technology, a lot of it has to do with bringing in radiation beams from many different directions.
Hmm.
And so, simplistically, if you think of like treating a small target, like a small round tumor, if you have radiation beams coming from many different directions, where those beams cross, the dose will add up, and in other areas, the dose is much lower. But it turns out that through computer optimization of the intensity pattern from each direction, we can actually build up this sort of focusing or sculpting of dose for much more complicated shapes.
Hmm.
And so then we can get, doses that sort of wrap around odd-shaped tumors. They could be convex, they can be
Hmm.
concave, and so on. And carve dose away from certain, critical organs that we want to protect, and so on. So that, the kind of fundamental technology there we call intensity modulated radiotherapy, IMRT.
Hmm.
And there's lots of different, flavors of that. Um, and then in addition to that, okay, if we can sculpt the dose and make it very accurately shaped, how do we ensure that it's actually going where we think it's going?
Hmm.
Right. And so imaging plays a role in that as well. Because so it's what we refer to as image guided radiotherapy. It's kind of incorporating image directly into the treatment process so that we can see, within the body where we're treating, ensure that the treatment plan that we're delivering actually is what we had planned up front.
Hmm.
And so having, for example, bringing three-dimensional imaging like CT imaging or
Sure.
MRI directly into the treatment process. We're taking these images, right at the time of treatment and making sure that the patient's body looks like what we think when we did the planning of the radiation. And then another critical piece is, okay, well, we can, if we're treating, a statue, we can create these very sculpted doses. We can do imaging and so on to make sure that the dose is going exactly where we're thinking that it's going. But we're not treating statues, we're treating living, breathing human beings.
Yep.
Um, and one of the wonderful but challenging things about living, breathing human beings is that we're always moving. Like every part of the body is under constant motion.
Yep.
You know, some more, some parts more, some parts less. So if you think about, one classic example, which applies to what I do because my clinical specialty is lung cancer. If I'm treating a tumor in the lung and the patient's breathing, that the organs are moving, the tumor is moving. And so I'm trying to do this very precise treatment to a constantly moving target, right? And so that's part of what my career has been about in within radiation oncology
Hmm.
is working with my colleagues in medical physics, um, developing techniques and technologies for what we refer to as motion management,
Hmm. Hmm.
ensuring that in this dynamic, moving, living body that the radiation treatment that we're trying to deliver very precisely is
Hmm.
going exactly where we want it to go and minimize the collateral damage to the surrounding areas.
Excellent. Excellent. All right. And again, Bill, I appreciate you laying the groundwork there because it feeds very nicely into what we're going to be talking about next in terms of Flash. And, here, just again, there's history here that the concept of extremely high dose radiotherapy goes back, I think, I was reading to the 1950s, but really didn't take off until sort of mid 2010, 2014 around where it started really picking up in terms of the literature on this. And as you were just mentioning, you as a clinician, you're looking for this very important shaping concept. When you do the radiation, you're looking for sparing. You want to hit cancer cells. You don't want to hit the good cells. And then what we're getting into, what you were talking about, motion management, and you put it very eloquently in one of your papers. This was in 2019 on the topic, this principle of freezing physiological motion. Um, take us a little into. I'm going to go into that one a little more because I think it's interesting. You were talking about the lungs moving. I was thinking when I saw that, like, wow, the other thing that our cells like to do, our cancer cells, if they get radiated, they don't like it, and they try to repair their DNA, and all sorts of shock proteins and this and that are produced. And that'd be cool to go into a little more. Talk a little bit about the history of Flash. And I mean, we're talking about it, and sorry, I had to do my Chernobyl conversion here. We're talking in these units of something known as a gray, which, I did my little Google thing, which goes, I think, 1 to 100 in terms of Ronkin, which, being a major Chernobyl fan, I know my Ronkins pretty well. But talk about what we're talking about here, because I was reading like a typical radiotherapy is only a couple grays per session, and you're going a couple orders of magnitude above that. Very briefly, with this technology, walk us through a little what we're talking about in terms of this very high dose principle.
Yeah. So thank you. You've touched on a topic that's obviously kind of near and dear to my heart, and is really the focus of where my research is. So FLASH refers to ultra rapid radiotherapy, and that there's kind of two aspects to that. There's a physical aspect, which is speed, and then there's a biological aspect, which is how do cells and tissues respond to that speed. Um, and so, and just to kind of frame it, in terms of units, as you said, gray is the unit of radiation dose that we use. And so it's the amount of energy that's deposited per unit mass. Okay. The units would be joules per kilogram. 1 joule per kilogram is a gray. Okay.
Okay.
Um, and and so that's kind of how we standardize radiation doses that we use for treating cancer. Um, it's really through historical, clinical experience that we've learned, certain tumors, require a certain amount of dose to have a good chance of controlling it or eliminating it. And so that's how, clinical care has evolved over time. So certain tumors, for example, you'd mentioned Hodgkin's disease earlier. That's
Mm-hmm.
a form of lymphoma that tends to respond very well to radiation, and so we use lower doses for lymphoma, and compared to solid tumors, as you called them, which refers to these tumors that are in forming in more solid organs as opposed to blood-based cancers. So lung cancer being, an example of that. Prostate cancer, breast cancer, and so on. These are all solid tumors, and they tend to require a higher dose of radiation. So a standard course of radiotherapy for lung cancer, for kind of a more somewhat advanced lung cancer, a larger tumor, would be, uh, just to give the sense of the units, a 60 gray.
Mm-hmm.
And that would be delivered over time. So
Yep.
that's delivered in sessions of 2 gray each time. So you might,
Yep.
give a daily dose of 2 gray and, 30 treatments over 6 weeks. That would add up to 60 gray.
Yep.
Okay. Um, when we're referring to flash, we're talking uh, not strictly about the dose, but the dose rate or the speed of the delivery.
Got it.
And so we might, for a given tumor, we might deliver, the same dose as we would conventionally, but we would deliver it in a much shorter period of time. And so to give you a sense of the time scales that we're talking about, a typical delivery of radiation therapy using, our current clinical state-of-the-art equipment, um, a 2 gray dose, which would be kind of the small daily dose, might be delivered over a period of maybe a minute or so.
Okay.
That's the actual like time that the radiation beam is on. If we're delivering higher doses, then it would take a little longer. It might take a few minutes, 3, 4, 5 minutes, something like that. Okay. And the regime that we're talking about for flash is to deliver those same doses in a small fraction of a second. So we're talking about a few hundred to maybe a few thousand times faster than what we can currently do with our conventional clinical technology. And so that gets back to the point, well, why is this relevant? Why, does this speed matter? And the way that, kind of I, uh, um, you know, uh, came into this story was going back to that angle of motion management, right? So I'm working with my colleagues to try to solve this problem of, it takes a few minutes to deliver this radiation treatment. I'm trying to shoot at moving tumors, like let's say a lung tumor. Um, and in that period of, a few minutes, the patient is breathing, that tumor is moving. And so how do I deal with that? What is the motion management strategy?
Mm-hmm.
So there's a few ways to deal with that. One is just to understand what is that motion that's happening. Historically, what people would do without having kind of better knowledge is they would just have to assume, well, I know that the tumor is going to move when I treat it. I'd better make the area that I treat bigger, so that I don't miss the tumor.
Mm-hmm.
But I don't know in this given patient how much it's going to move, so I can have to make some assumptions based on population averages and so on. And then I add some to that just to make sure I'm not missing. And then now I'm treating a pretty large area. Um, and yes, if I treat a big area, that will help ensure that I don't miss the tumor. But that also means that I'm treating a lot more normal tissue in there. And it's kind of it ends up at some point being counterproductive because I can treat a larger area to make sure I don't miss, but that limits how much I can give because the more normal tissue that I treat, the less dose I can actually give safely, right? So how do I improve that? Well, one way is to at least understand for a given patient how much motion is happening. What is that individualized, sort of tumor motion within that patient? And we can do that now with imaging techniques. We have what we refer to as 4D CT. It's just one of the techniques that we have. And so that's like doing a CT scan, but doing it as a movie. And
Okay.
so as the patient's breathing, we can actually see the trajectory of the tumor. And, some patients will have greater tumor motion, some will have less tumor motion. And then now I can take that into account. I know this patient, if the tumor is going to move, X amount, I can design my radiation, target to kind of match that amount of tumor motion and make sure I don't miss. But I don't have to guess. I don't have to just add on, an arbitrary amount. I can tailor it to that patient and make sure I'm treating, just what's necessary. Um, so that would be, what we can refer to as motion inclusive targeting.
Okay.
Um, but we can go beyond that. So we actually have the ability to synchronize the radiation beam with the patient's breathing. So rather than have the beam on continuously while the tumor is moving, I can I can turn the beam on, for example, only at the end of a patient's breath.
Ah,
Uh, and then, it's a much smaller area, or at least potentially a much smaller area. If a
right.
patient has a lot of breathing motion, then I can save a lot on that by treating only when the tumor is at a certain portion of the breathing cycle.
Got it.
So we refer to that as respiratory gating.
Hmm.
We can also do other things. We can restrict the amount of motion. So there's things like we refer to as abdominal compression, where we sort of put pressure on the patient's belly so they can't take in as deep a breath. Um, that's one kind of way to sort of force a less motion. Um, but we also have technologies to do tumor tracking, so we can actually follow the tumor around with the radiation beam. If we know where it is, we kind of shift the position of the beam to follow it. So we have technologies to do that. So these are all things that, I've worked with colleagues to implement in our own clinic at Stanford over the years. Kind of that's been one of our specialties is
Mm-hmm.
bringing these motion management technologies into the clinic.
Mm-hmm. Mm-hmm.
But as part of doing that, you know, there's a few things that we kind of insights that we gained and realized, um, as our technologies have advanced, gradually we've been able to speed up the treatment, actually pretty substantially. So I mentioned that treatments are, like a few minutes these days. Well, back, in, like early 2000s, around the time that I was initially doing my radiation oncology residency training, um, one of these kind of focused radiation treatments where we are trying to kind of compensate for motion and have the patient, hold their breath for a bit and treat and so
Mm-hmm.
on. Those treatments could take like a couple hours, even, 3, 4 hours sometimes. And so going from, 2, 3, 4 hours down to like 2, 3 minutes, there's already like a couple of orders of magnitude improvement in speed there, right? And what we find is that that has multiple benefits. I mean, we're not freezing the motion, but the less time that the patient is on the table, the less opportunity there is for, moving around, for just being uncomfortable,
Sure.
getting stiff, hurting and all that. The longer you're on the table, the more uncomfortable you are and the more you're likely to move, right? And so we saw kind of as treatment times decreased, we also got better and better at doing these motion management strategies.
Mm-hmm.
And the insight was, well, we're doing all of these things based on an assumption, right? There's a fundamental assumption here that we need to do all these very complicated, labor intensive, technology intensive techniques. And because they're complicated and because they're labor intensive, it takes a lot of expertise to do them. So it's not, easily, disseminated to the rest of the world to do this reliably, right? It's easy to do wrong. But it's all because of this assumption that it takes longer to deliver the radiation than for the body to move. And yet we're
Mm-hmm.
seeing this trend where, well, the treatment times are getting shorter and shorter. What if we could take that to its logical conclusion?
Yeah.
What if we could get the treatment times so short, like almost instantaneous,
Mm-hmm.
that now it's much shorter than the time scale of the motion. And now we're freezing the motion. Then during treatment, there is no motion, right?
Yeah.
That is kind of the ultimate motion management strategy is to eliminate the motion. And so it was sort of based on that insight that, my collaborators and I started focusing on uh, how technologically to approach very fast treatment, fast enough to freeze the motion. And so this was in kind of the early, 2010s or mid 2010s and so on that we were starting to work on this technology to develop very fast treatment.
Mm-hmm.
Um, but you brought up this point about flash, um, and so at that time, we weren't thinking in terms of flash. We were thinking in terms of freezing motion.
Yeah.
But in 2014, this publication came out of the Curie Institute.
Mm-hmm.
Professor Favadan and Vozenin led this study
Mm-hmm.
that showed something that seemed crazy at the time, right? Which is that in mice, they did these radiation treatments of the lungs of the mouse. And what they found was that, giving the typical conventional type of speed of radiation at a high dose, the lungs of the mouse get scarred down after several months, right? And that's a typical, well-known, reaction or side effect, if you will, of radiation to the lungs. High dose radiation to the lungs causes scarring.
Mm-hmm.
What they found was that, and this was possible to do in mice, giving the same dose of radiation extremely fast in a fraction of a second, which they called flash, the scarring of the lung was dramatically decreased. Um, and yet, when they looked at tumor models in these mice, the tumors, the killing of the tumors was not compromised, going to these faster speeds. So the tumor killing was equally effective, but the side effects were dramatically less.
Hmm. Hmm.
Um, and at first, I would say that that you know the that seminal publication has, of course, generated tremendous excitement and given birth to a whole new field of radiation biology research.
Hmm.
But initially, the uptake of that concept was kind of slow because people looked at that and said, first of all, is this even a real phenomenon, right?
Yeah,
It goes counter to like a century of radiation biology research. Um, and there's no explanation for it, mechanistically at that time. And what's the clinical relevance anyway, right? I mean, you can't give radiation treatments in a fraction of a second in a clinic. Of course, we were working on this technology to do exactly that, right?
yeah,
And so when we saw these results, we said, wait a minute, if this is real, then what we're doing will not have just this, kind of spatial precision benefit, but it'll have a huge biological benefit.
sure.
And so at that point, we really kind of went all in on the biological research side as well as the, kind of technology development side.
Hmm. Hmm.
Um, and so that's how I got involved in flash research.
Yeah. Yeah, let's drill down on some of that biologic research because some of the things that you're finding out, and I guess we can just start with some of the sparing issues. I mean, you've published again extensively on everything from radiation-induced GI toxicity in ovarian cancer mice. You looked at skin toxicities and ulcerations. Amazing comparisons. The one I'd love to focus a little bit more on, though, is this was October 2019, Journal Radiotherapy and Oncology, where you study the reduced impact on cognitive deficits following flash and looking at things like dendritic spine loss and neuroinflammation. I wanted to focus here because glioblastoma, unfortunately, has been a focus of our show. Not from this perspective, but clearly it's a pretty the nastiest of nasties out there. And again, substantially unmet need. You're finding really interesting results with, again, the ability to, quick flash radiation, but neural degeneration down, neuroinflammation down, cognition remaining very important with these patients. Talk a little bit about what you found when you were doing this work.
Yeah, yeah, yeah. So it is exactly as you said. What we found was that, and in my lab, the majority of the work has been on the normal tissue response side of things.
Yeah.
Um, and we have done some tumor models, and some collaborators of mine have done a lot of that. Um, uh, but what we have found is that, as I mentioned, generally speaking, dose for dose, if we give the same dose of radiation in the conventional way, conventional speed over like a few minutes, at a dose that causes, substantial side effects, the same dose given very rapidly in a fraction of a second or flash produces much less side effects. And so in the brain, what we have found is that uh, the normal, or I should say classical, expected things that we know radiation does to the brain is that it causes inflammation. It causes
Yep.
inflammation that lasts, long term. So we call that neuroinflammation. It's mediated by, many different mechanisms, but one of the key ones is activation of microglia, which are these immune cells in
Sure.
the brain, and causes an abnormal inflammatory response that lasts almost indefinitely after the radiation exposure. And that leads to many downstream effects, including alterations of neurons in the hippocampus of the brain, probably all regions of the brain, but the hippocampus is the most studied part of the brain where, it's key to memory processes. So there's alteration in the microstructure of the neurons. There's loss of these dendritic spines, as you mentioned. There's also loss of neurogenesis, so formation of newborn neurons in the brain. That's kind of critical to regeneration and memory processes in the brain, and that's lost after radiotherapy. But what we find is that, again, giving the same doses of radiation that would cause all of these side effects, if we do it with, flash, that the all of these effects are substantially reduced. That there's preservation of neurogenesis and microanatomy of the neurons, reduced neuroinflammation, all of these things. It's not zero. It's not magic, right? I mean, there's still,
Sure.
damage that's done, but and you can measure it, but it's substantially less. But the downstream effect of what it adds up to, like if you actually test the memory of these mice, and there's lots of fairly sophisticated ways of doing like behavioral and cognitive testing on mice. What you find is that, again, it's not like they got no, damage at all. If we drill down deep enough, we find some. But functionally, they are performing like mice who had no radiation. So that's, really remarkable and a dramatic finding.
hmm hmm And continuing along that, obviously you're involved in a lot of clinical development work, and one of the obviously we're in the era of immuno-oncology and everything from CAR T cells to oncolytic viruses and all this other cool stuff that is alive. And one of the hot areas in terms of Flash that I've read a little bit about are the excitement that obviously those living therapies don't like a lot of radiation, and the potential for using Flash in these combination therapies. I know you've been involved with other radiation technologies and immunotherapy. Thoughts on this? Because I think this is another important piece of sort of the clinical translation here, and how you can combine this technology with some of these other living therapeutics.
We were talking about synergy between radiotherapy and the immune therapies and the timing of the delivery of the radiation relative to when, critical immune cells may be, acting within the tumor. And so we have another lever when we can, adjust the speed of the radiation, another factor to optimize in terms of the timing. And so Flash may certainly impact that. And we do have some preliminary evidence that immune, responses or signaling can be different with Flash than with conventional slower radiation. So we did one study. This was a collaboration between the lab of my collaborator, Aaron Rankin at Stanford, and my lab, where we looked at in an ovarian cancer model in mice. We looked at the combination of radiation and immune checkpoint inhibition, which is one of kind of the current, it's really revolutionized a large part of cancer therapy.
Sure.
And what we found was that in combination with immune checkpoint inhibition, radiation produced the best tumor control given, when the combination was given, as opposed to any therapy individually. And this was true whether it was Flash or conventional dose rate. However, with Flash, the side effects on the intestines, the intestinal injury, was substantially less, even in the presence of the immunotherapy. So one question was, well, when you add something to the Flash, could you potentially spoil this Flash effect? All right. And that was an important finding, that the combination with immunotherapy did not spoil the Flash effect, that the normal tissue sparing was preserved. and we got this good interaction between radiation and immunotherapy. In addition, when we kind of drilled down to looking at the immune cell profiles in the tumors after radiation, whether it was flash or conventional speed radiation, that there was a somewhat more favorable immune cell profile with flash in terms of the ratio of T regulatory to effector cells.
Got it.
Now, that didn't actually translate into, at least in our study, into a difference in tumor responses, but there was at least a hint that there could be kind of a more favorable immune signaling going on with flash. Interestingly, a group at the University of Pennsylvania just published a paper showing something quite similar in terms of the effect of flash on these immune cell, kind of environment. And they found similarly that flash and the conventional speed radiation had about the same effect on tumor killing, but in the combination with CAR T cell immunotherapy, there was actually an enhancement of an anti-tumor effect with flash compared to the conventional speed radiation. So very early, results, but very exciting. I mean, I think there's just a whole, range of things to understand about the immune and radiation interactions, and potentially, mechanisms with flash that we can exploit that, are novel.
Billy, I neglected to mention at the beginning of the show that in addition to all your titles, you also have co-founded a company known as TibaRay that is developing sort of the next generation of flash technology. Again, I'm not going to talk about linear accelerators or Klystrons and Klystrinos. You talk about the most important parts of all this because there's a lot of really big terms here. I love the term phaser. I'm a big Star Trek fan, so I mean that I love that part about it.
Yeah.
What are you developing at the company? Obviously, it's the next generation of these accelerators of the radio frequency platforms. But talk about a little bit of what you're doing at the company.
Yeah, absolutely. Thank you for asking. So I mentioned that we had made this realization that what we really should be pushing forward for in kind of the next generation of motion management, and then ultimately in terms of flash, is high speed, uh, you know, orders of magnitude faster than what we can currently do with the technology we have in the clinic today. And the heart of the treatment machines that we have in our clinics is a linear accelerator. So this is a device that takes an electron beam and accelerates these electrons to very high energy. So they're essentially at the speed of light, and then convert that energy into an X-ray beam. So the linear accelerator is at the heart of that. And as it turns out, although we've had huge advances in our medical linear accelerator technologies over the years, incorporating, as I mentioned, more precision imaging, a lot of technology, the core linear accelerator technology itself actually isn't that different from when it was invented at Stanford in the 1950s. Only kind of modest evolution in that area. And we knew that that would be likely a very important core technology to tackle. So my colleague Peter Maxim, who's a medical physicist, and myself, we were working on this problem, and so we knew that we needed linear accelerator expertise. And that's when we got connected to my colleague, Sami Tantawi, who's a world expert in linear accelerator science and radio frequency power engineering. He was at SLAC National Accelerator Lab, which is, kind of a world-class accelerator science center that is at Stanford. And it happens that right at that time that we reached out to him, he and his colleagues had really made a breakthrough in understanding of the kind of fundamental physics of how linear accelerators work, where the power, the radio frequency power that goes into these accelerators, where does that go? We want that power to go into the beam to accelerate the beam, but most of it is actually going into kind of generating waste heat in the structure, right? I mean, every technology has its inefficiencies, and understanding uh, where that inefficiency was, what was causing it, and how to optimize the design to overcome it. That was kind of a key realization that a breakthrough that he had made at that time. So the timing was perfect for us to connect with him. And as it turns out, it was through I mean, he was doing this for a completely different reason, right? He is involved in accelerators for high energy physics research. For example, the discovery of the Higgs boson, at CERN, I mean, it came from an accelerator that can generate, an enormous amount of energy. But to explore physics beyond that, you need orders of magnitude higher energy. And the solution just cannot be to make an accelerator that's, as big as a city or halfway around the country. And, the power that would be needed for that is just not scalable.
Right.
And so his whole career has been about how do we make the accelerators more efficient, more compact, more higher energy for lower power, and so on, so that you can do these high energy physics, experiments.
Right.
And that was kind of the motivation that he came from. But when we came to him with, this problem we're trying to solve in cancer care, he said, well, this would be kind of the perfect way to realize, the benefits of all the discoveries that he was working on and have it, have a real, kind of a critical real world impact much more immediately in the lives of patients with cancer.
Mm-hmm.
And through that collaboration that we developed, actually that generated the grant funding to produce the first prototype, kind of proof of concept of these new physics principles that he had discovered. Um, and that's kind of what led to this concept that we refer to as phaser, the accelerator technology underlying phaser. And as you mentioned, we realized that to bring this to real world applications, that we had gotten to kind of an important stage within the university environment and scientific research, kind of demonstrating the proof of principle of these concepts, that the science is real. How do we make it real world? And we needed an entity to do that, and that's why we formed this startup that is called TibaRay. So TibaRay is where we are now, producing, kind of practical versions of this linear accelerator technology and other radio frequency power components that are far more advanced than the current state of the art. And that will be, the heart of how we translate this into patient care in the near future.
I was wanted to ask you about one other thing outside of oncology, because I noticed a couple months ago you published a paper in the archives entitled Rapid Sterilization of Clinical Apheresis Blood Products Using Ultra-High Dose Radiation. And it's kind of interesting because last year I had Dr. Najat Makhtar on the show. She heads up nuclear science at the International Atomic Energy Agency, and all she wanted to talk about was their zoonotic disease program. And obviously, radiation is pretty damn good at killing all sorts of nasty bugs and viruses and stuff like that. I thought this was extremely intriguing because clearly we need to sterilize all of our other important bioproducts, but yet we don't want to kill off the important cells and other biofactors that are in blood. And I would love if you could just say a few words about your vision for things beyond oncology, and in this case, sterilizing important human bioproducts.
Yeah, yeah, absolutely. So this was a collaboration with Dr. Ta Pham in pathology at Stanford, and kind of the director of the blood center, as well as Ed Engelman. And so they kind of came to us with this concept, that there's a need to safeguard the blood supply. Um, Blood products obviously are critical, for patient care, and there are challenges in this, right? You know, one of the big lines of defense against transmitting, infectious diseases through blood products is through screening of blood products. So it's just kind of ensuring that there are not specific infectious contaminants,
Mm-hmm.
but you can't screen for everything, right?
Right.
And having a way of sterilizing the blood products is important. Um, and so we looked at how do you do this potentially with radiation, which is used for sterilizing many things. It's
Sure.
used for sterilizing things like medical products, catheters, and other medical devices, and so on. Um, and in the industrial setting, that can be done if you have a large irradiator, at a central facility, and you can process and irradiate lots of, devices and materials and so on, or even food, things like that. Um, that's one model. But for blood products, that's much harder to do. I mean, really what you want is something that's point of care, right? Something where the blood center is, so that you don't have to ship the blood products, that you can, do it at the point where you collect the blood and distribute it and so on. And, the typical uh, irradiators that, kind of lab based irradiators that you have just don't produce radiation at a reasonable rate
Right.
to do the sterilization that you need, right? And so that's why there was a synergy here. We were developing accelerator technology that uh, could potentially produce much more radiation in a very compact form factor, and so it has the potential to be applied in this sort of use case where you want like a point of care type of sterilization with radiation.
Mm-hmm. Mm-hmm. Very cool. Um, Bill, one final question. I know this is going to go outside of the realm and into the theoretical, but I just wondering, and if it's the dumb question, just answer stupid. Uh, there's this phenomenon in biology that's pretty well established, known as hormesis, that, things that are harmful to us in high doses may have some benefit a low dose, from exercise to fasting to heat stress, whatever the case may be. I know the whole area of radiation hormesis has been kind of controversial in the past, whether any radiation is beneficial to human health and could stimulate repair mechanisms and resistance to disease. Thoughts on this topic of radiation hormesis and any potential benefit that you see in the future of phaser/flash technology, or is that just a wacky question and we'll move on?
No, no, I mean it's something that has been kind of discussed for a long time. Um, I'm honestly not an expert in that area. We're talking about like very low doses of radiation, and at very low doses, is all radiation harmful at any dose, or can there potentially be some benefits at very low doses? We know that at high doses, radiation is harmful to normal tissues, um, but we harness that for tumor killing. Um, as far as applications of phaser, I mean, our thoughts have all been around using it for, kind of the higher doses that would be used for tumor killing.
Right.
Um, uh, you know, I'm it's not immediately obvious to me the advantage of using that sort of technology to produce very low doses because you
Right.
don't necessarily need technology that can produce radiation very fast
Right.
to give these very low doses. But it's a really interesting area. But honestly, I'm not that familiar with it.
Yeah, again, I thought that if anyone's asked, it would be you, and that's cool. We'll put that topic over here. We'll talk about it a couple of years from now. But no, no, really, really amazing set of work. Just very impressive. While I have anything that I missed today that is on the horizon for 2025 in terms of upcoming clinical work, other parts, public-facing stuff related to TibaRay, anything I missed while we have your conferences, please.
Sure, yeah. Well, I think one of the big impacts that we want to address with uh, better radiotherapy technology. One aspect is more effective, safer cancer treatment.
Yeah.
But another aspect that's very important and kind of near and dear to my heart and those of my collaborators is radiotherapy is a pillar of cancer treatment.
Mhm.
And in the United States, about two-thirds of patients with cancer benefit from radiation at some point in their treatment course.
Mhm.
And the majority of those is actually with the potential, at least, and the intent to try to eliminate and cure the cancer. So
Mhm.
radiation is a curative treatment for cancer. Um, Obviously, we don't cure cancer as often as we would like. There's a long way to go,
Yes.
but many patients are cured, and many patients are cured with radiotherapy. But a big challenge that we face is because of the technological infrastructure that's needed. Um, Many patients around the world, in fact, about half of patients around the world who could benefit from radiotherapy have no access to it.
Mm.
And so we're talking about millions of patients today who, in principle, could be receiving curative potential treatment who have no access to anything other than purely palliative, you know, uh, uh, comfort care type of treatment.
Right.
So that's kind of a health crisis that's going on as of today. And if we can bring curative radiotherapy into a more compact, power efficient, economical, clinically efficient infrastructure, one that, for a given machine, we could treat many more patients um, at a given time, and make it portable and accessible. And that would have a huge impact and bring cancer care, curative cancer care, to places where it has, not been accessible. And, we think of places, for example, like Africa, where, I mean, there are 20 countries in Africa where there's literally zero access to radiotherapy. But to be honest, access to radiotherapy, even in the United States, in more remote areas and so on, just in terms of the logistics of distance to treatment centers and things like that, we have, an access problem right in our own backyard.
Oh, yeah.
So it's a huge problem and one that, with more efficient treatment that we feel that we can have an important impact. Um, I'll give an example, at Stanford, on one of our treatment machines, we do fairly complicated treatments at Stanford. And so, it takes sort of extra time and care to do all of the imaging and everything that we do. And so, um, on a given treatment machine, we may treat something like 20, 25 patients a day. Of course, we have multiple of these. Um, if we go to another place, for example, I've visited many centers in China, where they have, I mean, at their high-end centers, they have access to the same technology that we do. They have, very sophisticated machines. But just because of the pressing demand of volume there, they're using these same machines to treat like 80 to 100 patients a day.
Mm.
Um, and they can't do the same complexity and imaging and everything that we do, right? But if we had the technology, and this is again one of the visions of Phaser, integrated high quality imaging, instantaneous delivery, something that would allow us to treat. I mean, basically, we know it's possible to move that many patients through a machine in a day, but for all of them to be, treated with, the best quality curative treatment um, at that sort of throughput, that would have a huge impact on access to care and dissemination of the best cancer therapy around the world. So that's a really important part of the mission as I see it.
Yeah, no, it's an incredible mission. It's so impressive hearing about the arc of your career and obviously your vision for where this is all headed, and again this extremely important area of unmet medical needs. So, rooting you on. Hopefully, we'll do a follow-up episode a little down the line, Bill. But really amazing work. Again, for everybody that is going to be listening to this episode of the show across the various podcast networks or who will be watching on the YouTube channel. Again, you've been spending time with Dr. Bill Liu, professor of radiation oncology, member of the Stanford Cancer Institute, the Molecular Imaging Program, BioX, co-founder of TibaRay, principal investigator of the Flash Sciences Lab, doing really amazing things to bring this really transformative technology and clinically relevant technology to the masses. Bill, I want to thank you for taking the time out of your schedule to talk to us for a little while. Thank you for everything you're up to. And as we like to say here on our show, thanks for helping to create a better tomorrow for millions of patients. Awesome story. Awesome
Thank you
story.
so much.
Really great having you.